This unit is going to deal with immunology. Immunology is an up-and-coming science that deals basically with antibodies and antigens and how we can recognize them. An antigen is any chemical that is recognized as non-self and that triggers an immune response in a mammalian system. An antibody is a Y-shaped protein that is created by an organism and that binds to antigens. These have a typical Y shape. It's got 3 edges to it. 2 of these edges are called antigen binding sites, which is where the antigen will bind to. These have lock and key specificity, meaning that they only work on one particular antigen, and therefore you have many different kinds of these in your system. They are all produced by B cells. And when we deal with them, nowadays we deal with something that are called monoclonals, and we'll get into that in a minute. When we deal with antibodies, we call these serological tests because we normally used blood serum that has antibodies or antigens in it. We have direct tests, which test for the presence of antigens. In a test like this, we might be looking at something such as, do you have the organism that causes strep throat, or is salmonella present in a specific food supply? And we're testing for the presence of antigens. In indirect tests, we're testing for the presence of antibodies. Many times they ask you, have you had chickenpox, and how strong is your reaction to it? Because if you don't have a strong enough reaction, you're going to have to be revaccinated. You would do an indirect test to test for the presence of antibodies in your bloodstream to find out how many antibodies you have. We have qualitative tests. Qualitative test says it tests for the presence or absence. This is like a pregnancy test. It's a yes/no. Either you are or you're not. We also have quantitative tests. Tests for the amount. Again, if you go back to the chickenpox example, when you get tested, you are looking for how much do you have. Now, we start out over here with a 4-legged furry creature. The amount of antibodies that you needed determined the size of the 4-legged furry creature. In the past, they used to use some rather large ones. This got to be expensive because you had to have the organism in a sterile environment. Nowadays, the procedure has changed. What they do is they take the 4-legged furry creature, they inject it with antigen, its immune system reacts to that, and then they are able to do a splenectomy and remove the cells that produce the antibodies. These then get fused with myeloma cells that are grown in a cell culture, and what we get is we get something called a hybridoma. The hybridoma has the longevity of the myeloma cells, and it also has the ability to secrete the antibodies. These are then plated out so that we can determine exactly which one is producing the antibodies that we want. We can then take that particular material and we can grow that particular material in a cell culture, and now all day long we have a constant supply of new antibodies being created by these perpetual cells. So we get quality control and we get much higher levels of production. Another test method we have is what we call a diffusion test. Now, we start out with an agarose material that is a very thin type agarose. It's 0.1 to 0.2%, which is much thinner than what you find in culture media. So it's kind of like a solidified water. We place the material that we are trying to test in these little wells that we have perforated in the agar. And what will happen is the material will diffuse out. When it comes in contact with a compatible material, you will get a line of precipitation forming, indicating the presence of the antigen and antibody for that particular test. When you look at this, you can see the 2 lines of precipitation in here as we are able to complete this test. Another type of test we have is called the ELISA test, or the enzyme-linked immunosorbent assay. Test is rather simple. We start out with a microtiter plate. We place into it our antibodies that are for the specific antigen that we are looking for. These will hook onto the bottom of the plate. We then rinse out any material that has not attached in. We add our sample. If the sample has the antigen present in it, the antigen will bind onto the antibodies. Again, we rinse. Then we add another antibody that is just like the first one, but in this case it has an enzyme linked onto the end to the antibody. If the antigen is present, these will bind onto the antigen. If the antigen is not present, these will not bind on. The amount of antigen present determines how many of these actually get bound. We then take that plate and we add to it a substrate that will react with the enzyme, and that will cause the substrate to break apart into the product. The product will give us a color development. The intensity of the color will depend on how much enzyme we have present in this. When we look at this particular plate, you can see that we have various colors, and this shows the development of colors at different concentrations of antigen so that we can actually see color development. Again, this is qualitative because the darker the color, and you can actually measure these colors, the darker the color, the more antigen had present. This is a dipstick type test. We can use this for a lot of different materials. In this particular case, this is a plant material. Our sample is placed on the inside. The sample is macerated, and that is going to release the contents of the cells. You take your dipstick and insert it in. The material is pulled up on the inside, and what you get is a result like this. The upper one has a single band, therefore the answer is no. The bottom one has a double band, therefore the answer is yes. This is the type of test that we have for things like strep pyogenes in the throat, as well as this particular one, which is looking for a pathogen in a plant sample. A pregnancy test works in the same manner. This is an agglutination test. And in agglutination test, what you're trying to find is whether or not the material is present. And what it does is it forms large particles, and the large particles come together and they clump and they fall out of solution. This particular one is a blood test, and we do blood testing in order to determine different antigens that are present on cells. So when we look at immunological testing, there are a lot of different possibilities that we have, and they are all made possible by antibodies that are produced in mammalian systems that we can use to test for specific antigens.